In vivo FPOP probes protein interactions and protein structural changes in C. elegans. These animals are widely used as a model system to study human disease.
By coupling in vivo FPOP to mass spectrometry, we can probe protein-protein interactions within different cellular and body compartments in a live animal without the need of isolating one particular protein.
Begin the flow system assembly by cutting a 2 cm piece of fluorinated ethylene propylene, or FEP, tubing. Use a clean dissecting needle to widen the inner diameter at one end of the tubing, creating a small crater about 50 mm in length. To create the infusing lines of the flow system, cut two 15 cm pieces of 250 micrometer inner diameter fused silica with a ceramic cutter, and tape the two capillaries together with self-adhesive tape, making sure that the ends are 100% flushed. Insert the two capillaries into the crater of the FEP tubing, pushing them up to the very edge. Place a small dot of epoxy resin on a clean surface and mix it with a dissecting needle. Use the same needle to quickly place a small drop of the resin at the end of the infusing capillaries where they connect with the FEP tubing. Allow the resin to dry, outlet side up, for a few minutes. Meanwhile, cut a new 250 micrometer inner diameter capillary, which will become the outlet capillary of the flow system. Once the resin has dried, insert the new capillary through the FEP tubing outlet end. The inside ends of the outlet capillary and the two infusing capillaries should be flush against each other, creating the mixing T. Bind the capillary and FEP tubing with fresh epoxy resin as previously described,